is Delaney. Today I'm going to be doing my first anatomy lab report. It's going to be about the microscope, and as I do the simulation, I'll be taking you with me, and yeah, we'll learn together. Hello. Before we get into the simulation, we'll quickly go through the different parts of the microscope. As I write them down, I'll have the definitions appear on the screen as well. Okay, so getting to the microscope, it's important to know how it works. This simulation is definitely very different from the real microscope, but it's a good way to understand how it works anyways. First things first, when using the microscope, you want to start with the lowest power lens, which in this case is the 4 times power lens. When you start in your real microscope, the light power should be off, so start by turning it on and increasing the light. In this simulation, not much had to be adjusted since it was already on. Next, you want to use the coarse focus to get the best view of the specimen viewed. Make sure to always have your specimen properly positioned in the center, otherwise you might lose it as you progress. You may adjust the light, but for the lowest power lens, stick to the coarse adjustment and you can progressively adjust the find as you go along. Here is what it should look like. Moving on to the next power, now you just want to use the fine adjustment to get the best focus of the specimen. Adjust the light and again position the specimen properly in the center of where you want it viewed. Continue the steps of adjusting until you reach the highest power, or also known as oil immersion lens. This lens has the highest power and requires a few drops of oil to be able to see the image clearer. While in this power lens, make sure to not use the coarse focus, because doing so will cause the objective lenses to possibly damage or crack. This is a very simplified process of using the microscope, but with these few principles in mind, using the real microscope shouldn't be too different. And of course, like the real microscope, cleanup is important. Use lens wipes to wipe off the oil. Carefully return the objective lenses to its original position, which was the lowest lens first, and put the stage back down. These steps will allow you to keep your microscope lasting a lot longer. In this video, I learned about the orientation of the specimen viewed. It's important to remember that images seen in the microscope are upside down and backwards due to the mirrors a microscope contains. In the second video, we learned how to make our own wet mount of cheek cells that we can scrape from the inside of our cheeks using a toothpick. The process was simple and consisted of the usage of just methylene blue, slide with a cover slip, water, toothpick, and a tissue paper. In the third video, the main concept I learned is depth of focus. I learned that as the magnification increases, the depth of field decreases, and so does the depth of focus.